Hi there, I'm Jen Harrell, a biology professor at ACC, and I'm here today to talk to you about Lab 3B, Introduction to the Microscope. 1406 biology is cellular and molecular biology, so it should come as no surprise that you're going to spend some of your lab time looking at actual cells. In Lab 3B, you're going to learn the parts of the microscope, how to use the microscope and care for the microscope, and you'll also learn to use a couple of different types of specimen slides. This is the type of microscope you'll use in lab, a bright field, compound, light microscope. Now all microscopes magnify images, right? That's what they do. They make small things look large. So how does this particular type of microscope accomplish that? Well, let's break down the name a little bit. First of all, it is a light microscope. So it's going to pass a beam of light from below through the specimen. And then that beam of light is going to travel through a series of lenses which magnify the image before it hits your eye. Because this microscope uses more than one set of lenses, we refer to it as being compound. This first set of lenses on the bottom, these are known as the objective lenses, and they magnify the image a certain amount. There are different objective lenses that can be used. And then the lens in the eyepiece up here is known as the ocular lens, and it typically magnifies the image another tenfold. It's a 10x lens. So the image is magnified more than once before you actually get to see it. Now this type of microscope is also known as a bright field microscope. That refers to the type of image that's produced. Because a beam of light is passed through the specimen before it hits your eye, bright field microscopes typically produce dark images on a bright background. Now this restricts the use of this type of microscope a little bit. In order to get a good image on this type of microscope, the specimen you're looking at has to be extremely thin, or the light won't pass through it correctly and you won't get a very clear image. To simulate what it's like to use an actual microscope, you'll be using this website, the NC Community College Virtual Microscope. You can access this page by clicking on the link at the bottom of this slide, or also on the link in your protocol. Once you access the web page, your screen should look approximately like this. Now, carefully following the steps outlined in the protocol for the Lab 3B, make sure you read through that lab and follow the steps, you'll do the following. First, you're going to learn about the microscope by accessing the guide at the bottom of the page. This will give you a little bit of introduction to the equipment. Be sure to click on any little blue arrows that you see to proceed all the way to the end of the lesson, or you'll end up skipping some stuff. Once you've gone through those slides, you'll click Close at the top, and then you'll go into the Learn tab. The Learn tab is going to quiz you a little bit and also give you an opportunity to practice what you know about the parts of the microscope. For instance, it will teach you a little bit about these ocular lenses, also known as the eyepiece lens, the various knobs, the base, and the stage manipulator controls. Once you've worked through all of these portions, it will allow you to proceed to the next part of the demonstration and do work your way all the way through the demonstration. When you're finished with the Learn section, you can click Main to go back to the main page. Once you've worked through all of the introductory material as specified in the protocol for your lab, you'll be ready to actually use the virtual microscope. To do this, you'll click on the Explore button at the bottom of the page. Next, you'll select a slide. The slides are located in this gray slide box, and you'll see that there's a variety of slides available. You'll begin with the sample slides and the letter E. Letter E is just a little piece of newsprint that's been affixed to a slide, and it's designed to help you practice with the microscope, just learning how to focus it and use the microscope. Again, following the instructions specified in your protocol, you'll then begin to work the microscope. In general, this is how microscopes are focused. First, you will select the smallest power objective lens, which in this case is the scanning lens, the 4X. Then you'll start by adjusting the coarse focus knob. This is going to be a larger knob on the side of the microscope, and you'll notice that as you adjust it, the image in the field of view, the circle that you're looking at, becomes clearer. And you'll also notice that the stage on the actual microscope moves up and down a lot. This is the reason that we only use the coarse focus adjustment when the microscope has the smallest lens, the scanning lens, engaged. If you try to do this with a higher power lens, you run the risk of poking the lens into the slide itself, and you can damage both the slide and the lens itself. So coarse focus should really only be for 4X. Now once you have your image focused about as well as you can with that focus knob, then you'll move over to the fine focus knob. And with this you fine-tune your image as best you can. Notice that when you move the fine focus knob, the stage does move a little bit, but not nearly as much as the coarse focus knob. So this focus knob is safe to use with any power lens. Once you've adjusted your fine focus as well as you can, you might also want to play with the amount of light. Now in this case, because we're on a lower power lens, you notice that more light is not necessarily better. But as you proceed through the lenses and you get on higher and higher power lenses, you will notice that the aperture of the lens is smaller, therefore you're going to need more light to get a good image. Once you've adjusted your focus knobs and your light, you can then move up to the next power lens. Again, once you're on a higher power lens, you'll only want to adjust the fine focus. You may also notice that the slide has moved around, so you can't quite see maybe the part that you wanted to see in the first place. So you can move the slide around by clicking on the field of view and clicking and holding and dragging it around. On a real microscope, there are stage adjustment knobs that allow you to do this, but of course you can't adjust those with the virtual microscope. Once you have the image the way you want it, you can move up through the higher lenses and magnify it even more. Notice again, I'm on a higher power lens here. I'm going to need a little bit more light to clarify that image a little bit better. The 100X lens is also known as an oil immersion lens. We really only use it for looking at things that are very, very tiny, like bacteria. So you'll use it once in Lab 4, but that's about it. So we're not really going to worry about learning about this particular lens too much. For the purposes of almost all the specimens you'll look at in this class, we will use the 4X, the 10X, and the 40X magnification. This slide provides the steps for how to properly focus a microscope. Again, you only want to use that coarse focus knob when you're on the lowest power lens, the scanning lens. Now I know you couldn't see it very well on this virtual microscope, but this is the coarse focus knob, the big knob here on the side. And again, it moves the stage up and down quite a bit. The other knob that's available is the fine focus knob. And again, it's safe to use on any power lens because it only moves the stage a tiny bit. But it's typically used after the coarse focus knob has already been adjusted on the lowest power lens. This video is a great demonstration of the proper use and care of a microscope. Watch the video at the link provided. This link is also available in the protocol for Lab 3B. It's very important to properly record any observations that you make while you're using a microscope. So let's talk about how you might do that. First of all, notice this circle over here on the side. The circle that you see when you look through the microscope is known as the field of view. And so it's a good idea to actually draw the field of view on your paper as you're making your drawings. That's going to provide the observer, anyone that's looking at your records later, with a little bit of perspective on what exactly you saw and how large it looked through the microscope. But that can provide some important information. So draw your field of view. Next, you want to make your drawing inside your field of view. You want to make it as detailed as possible, but you don't have to be a great artist to make good observations. It's a good idea to include color and to include as much detail as you can. Once you've got your drawing of what you actually saw, you'll want to provide a little bit of information beneath the field of view. First, you want to provide the name of the organism, if you know it, or if you don't know it, you could say something like unknown protist. Second, you want to provide the total magnification beneath the name. The total magnification represents how many times the image you are looking at was magnified from the original. It gives the observer a little bit of information on how large that organism is relative to how much it was magnified. To calculate the total magnification, you want to take the power of the objective lens you were using and multiply it by 10. You have to do this because remember, this microscope is a compound microscope. So the light, the image, passes through two sets of lenses before it hits your eye and thus is magnified twice. So if you were using the scanning lens, as in this case, the objective lens is 4x, magnifying it 4 times. The ocular lens is then going to magnify that image a further 10 times. So you multiply together the power of the objective lens times the ocular, which is always 10. And that gives you a total magnification of 40x. In other words, the image you are viewing is 40 times larger than it actually is. You'll work with two different types of slides in lab: prepared slides and wet mounts. First, let's talk about prepared slides. As the name implies, prepared slides are already prepared for you. With prepared slides, the organism of interest is already fixed to the slide, so it's glued down in place. There's a cover slip that's already glued on top of it, and the slide is labeled with helpful information about the identity of the organism and a little bit about how it was prepared. Prepared slides are really nice for looking at organisms without them moving around. So the organisms are dead, and oftentimes the organisms will be stained. You can see an example of that in some of these slides here to the left. Keep in mind that most cells and cellular structures are clear, so when you put them on the clear background of a slide, it makes them really, really difficult to see. So often with prepared slides, stains and dyes are added that will stick to certain cellular parts depending on the dye that's used and will make them a lot easier to see. The other type of slide that you'll use in lab is known as a wet mount. As the name implies, wet mounts usually contain something wet. So for instance, you might collect a little dust off the floor and combine it with liquid to visualize it under the microscope. Or you might add a drop of pond water or a leaf to a slide with a drop of water, or maybe even a drop of your own blood to a slide. Wet mounts are useful for seeing live cells. So oftentimes if there are cells present on the slide that can swim around, you'll get to see them moving around. They're good for observing behavior. They're good for observing movement and also relative size. You can kind of see things compared to other types of cells. Wet mounts are made by placing a drop of liquid on top of the specimen and then placing a little piece of glass on top known as a cover slip. The cover slip compresses the specimen, which makes it thin enough to pass that beam of light through, and also helps kind of hold things in place. Now there is a particular technique to making a wet mount. There are sloppy ways to do it that will not give you a good image, and there are proper ways to do it that will give you a good image. So in the next slide, you're going to learn how to make a proper wet mount. This video gives you a good set of instructions on how to properly make a wet mount slide. So go and watch it. The link for this video is also available in the protocol for Lab 3B. Alrighty, once you've gone through all of those careful steps in the Lab 3B protocol and played with all those nice settings on the virtual microscope, you should be pretty familiar with the proper care and use of a real microscope. Be sure you do this before you tackle the next lab, because you're going to use that microscope pretty extensively in this lab and a little bit in the following lab as well. Have fun. We'll see you in the next video.